professor in the Department of Oral Health here at UW, and he's going to be telling us about geroscience and oral health. So, John, good to see you, and go ahead and take it away.
Thanks, Mitch. I'm just going to get this set up here.
Oh, yeah, and I'll have a little 1 minute thing to let you know at 1 minute, so just keep an eye out.
Cool. Let me share my screen. Okay, great. Screen looks okay. All right. Well, thank you again, Mitch and Alessandra, for inviting me to talk today. So my name is Jonathan Ahn. I'm currently in the University of Washington School of Dentistry. So I'm a dentist who studies the biology of aging in the context of oral health. Why I became interested in it, I think I have to go back to kind of where I kind of came up with this idea of applying geroscience to oral health, and that was actually in the clinics. So when older adults or elderly patients present to us in the clinics, not only do we look at their dental health, but we also monitor their medical health. And many times, or elderly patients come with medications, one or even more, as described there, and this becomes very critical because it impacts the treatment or dental treatment that we render. But also a majority of these drugs that our patients take are a result of some form of chronic condition or diseases that they have, whether it is heart disease or diabetes. And we always tell our elderly patients to, or even older adults, to come in frequently so we could look and maintain their oral health. And so I'm just going to give you kind of a clinical example of where kind of age-related dysfunction and decline kind of impacts our treatment care. So I'll be showing some clinical photos. So for example, about 20 to 30% of older patients that come to the clinic have low saliva or dry mouth. And again, this is just one particular example. And saliva is important because it has important elements relating to buffering capacity. So they have calcium and phosphate to neutralize plaque acids or bacterial acids. But it also helps kind of clear the leftover food that kind of stays behind. And what happens is if you have low saliva or dry mouth, you have what's called plaque accumulation. So that's kind of food that gets left behind. And you could already see kind of on the image itself that plaque accumulation leads to kind of gingival inflammation. And so something like this kind of continues, it leads on to something like periodontal disease. But the example that I want to give is actually dental cavities or caries around the roots. And so what happens is once the hygienist or dentist cleans off that plaque, we find presentation of cavity. And so as a dentist, we go ahead and restore those, or we fill it, and then the patient leaves, 6 months to a year. But then because they have low saliva, they come back, they have this plaque accumulation, again, root cavities, or sometimes what we call recurrent cavities, or cavities again that happen in the same area because of the plaque accumulation. So as clinicians or as dentists, we're still treating the symptoms of this decline, and this becomes a cyclical process. What we should be actually doing is looking at the causal mechanism of this, which is low saliva, and which itself is actually induced by age. And so, and this is the same for periodontal disease, where when patients present, they already have some form of significant bone loss or bone inflammation. And again, we're still treating the symptoms rather than the actual cause, which is age. And so this is why, as a dentist, it's almost very clear to me why the geroscience hypothesis fits, in that in order to look at these various oral diseases that happen, we cannot look at them at an individual level, but by looking at it in terms of understanding the biology of aging and potentially targeting that process, we might be able to, what I call, extend the oral health span, or make the oral health conducive to health in older patients. So today, what I want to kind of describe are kind of one of the first projects that kind of helped me bridge kind of these two interfaces of oral health and aging biology, and then also kind of wrap up with just some preliminary data that we have in terms of another organ that we're looking at right now. So I want to first talk about the impact of rapamycin on periodontal disease. So periodontal disease is a bacterially induced chronic inflammatory disease. It is a disease of the periodontium, which is the substructure underneath and next to the tooth. So it includes not only the tooth itself, but the gingival or the gums, as well as the bone. And actually, it's the change in the pathogenic to pathogenic microbiome that causes this host response to fight off that infection. And obviously, because the microbiome is attached to the tooth, the tissue just continues to inflame. And when the inflammation continues to occur, that leads to bone loss and further inflammation until you could go see your hygienist or dentist to get the teeth cleaning done. And in fact, patients over the age of 65, roughly about 67% will have some form of periodontal disease within their mouth. And so currently the clinical definition of how we look at periodontal disease, and this is a definition from the American Academy of Periodontology, is that it's an infectious disease that results in attachment loss, inflammation, as well as bone loss with a change in the microbiome. And so when your hygienists take these x-rays, we're not only looking for potentially old, new cavities or making sure all the other components are still there, but also looking at bone loss around the teeth and seeing and making sure that those are maintained. And so when I first started my graduate study, I really wanted to kind of take this project and within the laboratory. And the laboratory I was in, they were looking at three different model organisms, at least yeast and then worms. There was also flies, and none of them had teeth. So I naturally went with the only model that had teeth, which ended up being mice. And so the first thing I did was I quickly tried to develop an assay to look at periodontal bone loss. And so I ended up using this high resolution micro CT imaging, where I went ahead and kind of teased apart the actual mouth. And what I found was that actually just age alone, so these are not artificially induced models, is that in both the top teeth or the maxilla or the bottom teeth or the mandible, that just with natural course of aging, that these animals or mice, these mice actually develop periodontal bone loss. And so in addition, I kind of utilized some of the clinical applications that we currently do for humans and kind of created the system where we're able to measure the distance between certain anatomical landmarks to really get a measurement. So this is kind of in pseudo of what your hygienist or dentist will do when you go to the dental office and they look at those pockets. So those values like 2s and 3s are kind of what they're measuring of those pockets. So this is a way to do that in mice. And so with this assay established, we've decided to look at whether or not a geroscience intervention was impacting this. And so we looked at rapamycin. And so rapamycin is a specific mTOR inhibitor. mTOR is a growth factor, a nutrient responsive kinase. Rapamycin was initially developed as an antifungal agent, but it has a potent immunomodulatory effect. The fortunate thing was when we were asking this question was that a lot of researchers were collecting the mice, but they really never looked at the teeth. And so before we kind of put resources into developing a new project, we wanted to look at kind of projects that were already completed that utilize different rapamycin dosages and seeing if the hypothesis that rapamycin may impact periodontal bone loss is valid. And so fortunately, we were able to kind of contact Peter and Ellen here at the University of Washington, who had just finished up a study looking at an 8-week treatment course in old animals at 14 parts per million of rapamycin, and this is given within the food. As well as we were able to get in contact with Rich Miller, who had these UM-Het3 strains. So these are four-way crossed mouse strains that were given lifelong treatment of rapamycin at 42 parts per million. And these two dosages, and this is just publication from the study that's showing that in females and also in males that there was an extension of life with increasing rapamycin dosages. But because rapamycin had this greater effect in females, we first started off looking at female mice. And so, again, these were all kind of stored in the lab. I went ahead and picked them up and isolated out the jaws. And basically what we found with the 14 parts per million rapamycin, not only, so on the right, you're seeing kind of our assay where this increasing distances is showing an increase in periodontal bone loss, that with just 8 weeks of 14 parts per million rapamycin treatment, there was this attenuation of periodontal bone. In addition, when we then looked at these UM-Het3 strains that were given lifelong treatment, we also found a very similar phenotype where, again, the control animals, you have severe periodontal bone loss around the teeth and in between the teeth, but with rapamycin treatment, there was this attenuation. So in two different mouse strain backgrounds, we found that rapamycin was having this attenuation effect on periodontal bone loss, which gave us a good reason to kind of set up our next experiment. Because this was the first time to show impact of rapamycin, at least in the context of aging, looking at an oral disease, we wanted to make the study as robust as we can, and we could have done more. But so what we ended up doing was raising two different colonies of mice. So some of the colonies were raised here and completed the treatment here in Seattle, Washington. We had another cohort of animals in Bar Harbor, Maine at the Jackson Laboratories. And so these were also aged to the respective ages, and then they were given 8-week treatment with either control or rapamycin. And then we harvested the animals after the 8 weeks. For the Jackson cohorts, we also implemented a longitudinal assessment where we took an initial micro-CT and then a final micro-CT to look at the same animal before and after treatment. And so how we approached analyzing data was really just strictly from a clinical approach. We wanted to look at the microbiome changes. We wanted to look at gingival changes as well as bone. And so the first tissue we looked at was gum tissue or gingival tissue. And obviously in humans, we could quickly, as a dentist, I could quickly look at it and whether or not determine there's inflammation, but it's a little bit challenging in the mouse tissue. We know that the gingival epithelia or the epithelium is one of the first line of defense against the microbiome, airborne pathogens, food, including even the masticatory process. So as you're chewing, think of the damage that it causes and is able to maintain homeostasis. So as a barrier site, the immune system needs to create this fine balance between surveillance as well as immune response to maintain that homeostatic environment. And then actually, when we look at kind of cytokine panel in terms of just the gingival tissue, so this is based upon a protein blot assay where we're looking at inflammatory cytokines and chemokines, is that relative to the young, which we have set here to 1, the old animals, the gingival tissues have increased inflammation here shown in the pink or the magenta, and then rapamycin treated animals, the gingival tissues show a decrease in the inflammatory inflammation and cytokines. And in fact, if we look at the protein complex that controls a lot of these cytokine production via NF-kappa beta, we see that there's an age-related increase in P65 signaling, and that rapamycin is able to attenuate that response in the gingival tissues. The next thing we looked at was bone. And so a bone turnover is a fine balance between bone formation as well as bone resorption. And one of the markers we actually looked at was for bone resorption or wrinkle. And multiple papers have shown that if you look at human periodontal disease, that this wrinkle expression not only is increased in the bone, but also in the gingival fluids. And so when we looked at wrinkle expression within the periodontal bone, we find that again, even in adulthood, there's an increased production of wrinkle or increased expression of wrinkle, but rapamycin was able to attenuate that. And similar to the gingival tissues, we find a variety of inflammatory related cytokines and chemokines. Again, this is just age related to be increased relative to the young animals, and rapamycin is able to suppress that or attenuate those responses. When we look at the phenotype of this, we find that rapamycin is able to attenuate bone loss. So on the left, you're looking at an adult mice and then old mice where you clearly see the periodontal bone loss happening, and that rapamycin is able to attenuate that. And this is the top teeth or the maxillary teeth. The bottom is the mandible. So again, you see an old mouse and an adult mice and an old mice. And then the rapamycin treated animals, you can see where there's some areas where there seems to be bone. Again, this is not longitudinal. This is cross-sectional. And so overall in our cohorts here in Seattle, we find that rapamycin was able to attenuate the periodontal bone loss as well as have less gingival inflammation and as well as periodontal bone inflammation. When we look in the Jackson cohort animals that we started, we again see this increase in wrinkle expression in the old animals, and rapamycin was able to attenuate it. And the real neat part about this is that we were able to look at the same animal before and after treatment. So here I'm just showing you a control animal. So on the left, you're seeing kind of the animal biting down, but you could see the periodontal bone loss in these old animals. And then after 8 weeks of just control, there's not much difference. There might be subtle differences where there's a little bit more bone loss, but again, it's very small. When you look at the animals, again, the same animals before treatment, you again see the periodontal bone loss between the teeth as well as between the roots. And then in fact, after just 8 weeks of treatment with rapamycin, what you're seeing is this kind of deposition of bone in those areas that were once missing, and including even in the mandible samples where again, you're seeing bone. This is just another animal showing you loss of bone between the two roots, and this is just kind of where we angled it towards you just to see, just to show you the bone line. And again, after just 8 weeks of rapamycin treatment, we see that there's deposition in there, bone deposits. So overall, even in the Jackson colonies, the same animal before and after, what we found was that there was this regeneration of the periodontal bone. And one of the questions we asked was, there was more bone, but is this bone really good? So what is that bone quality? How is that bone quality affected? You could think of osteopetrosis where there is more bone, but the bone is very fragile. So to do this, we kind of got creative, and we actually collaborated with mechanical engineering here at the University of Washington. So this is property mapping, and this is usually done with dental materials to look at how much hardness or elastic modulus there is. And this group was actually able to develop an assay to look at biological structures. And so hardness is basically, you're looking at the material's resistance to deformation, and then elastic modulus is the ratio over stress or strain or stiffness. And so what they do is you're able to quantify in a 10 by 770 micrometer area where a constant force of 200 micronewtons are applied, and you get a measurement of the hardness and modulus based upon that. And so we looked at this furcation area, and what we find is, and this is just the initial pilot study that we did, was that in the old animals within the furcation, we find that there is a significant decrease in both hardness and modulus. And when you actually look at similar property mappings in femurs of old animals, you find the same result. And then when we completed our study, we went ahead and submitted samples, including the rapamycin. And what we found was that there was indeed a decrease in the hardness and modulus, but the animals treated with rapamycin also had an increase in hardness as well as modulus. So the bone quality, there was an increase in bone quality after rapamycin treatment. The last part of periodontal disease is the microbiome, and we looked at it in collaboration with Jeff McLean and Chris Kearns here. And so when we first looked at alpha diversity, so you're looking at how many different species there are within a system. And so basically what we found was that there was a change between young and old, as well as what we find is that there seems to be this reverted shift of the old microbiome to be more similar to the young microbiome. And so what you're looking at here is a principal coordinate analysis showing the young animals here in turquoise, the red in the old, and what you see is the rapamycin treated animals are overlapped with the young. So there was this reverted shift. So that's just my conclusion. I'll go ahead and take any questions from that. Thank you again, Mitch, for the for the thing there.
Yeah, absolutely. Thanks, John. Super cool talk. So we've got time for just one quick question, I think. Yang Pan has an awesome question. So does rapamycin administration have to be systemic, or would something like a mouth rinse do the trick?
No, that's a great question. And that's something we're looking at. So our lab right now is doing what we call localized microinjections within the mouse tissue, in the gum tissue, to see if that replicates the systemic response. We don't know, but